My stepfather-in-law, I'm not an expert in video production, but I did the best I could. Okay. All right. So we'll go ahead and we'll get started today. Okay, so today we're talking about nucleotides and nucleic acids. So we have 2 lectures devoted to nucleic acids. All right? And so different professors will put different emphasis on nucleic acids and emphasize different things, especially because this is the part of the course where we really start to overlap with molecular biology. But as biochemists, we're really interested in structure function. So today we'll be focusing on, okay, so it's called nucleotides and nucleic acids because we're going to be focusing on drawing the nucleic acids, figuring out how they hydrogen bond, what happens if you swap out, something happens to a nucleic acid, it has a different identity, how's that going to affect the hydrogen bonding, and how's that going to extrapolate to the whole sequence of the DNA, and by extension, the sequence of any protein or gene or regulatory mRNA that's going to come as a result of that altered sequence. Yes, Sydney? Nucleic, this is the first lecture that will be for next test. Yes, because I don't want you taking a test on something before you take the structure quiz. Yeah, and this is like the weird thing where the structure quiz is a month from today, just because the way spring break and everything fell. It's not ideal, but yeah. And so the structure quiz, I don't have it up here yet, but the structure quiz is the 28th of March. Okay, so, and then we're gonna commence where we're going to think about nucleotides as how they get incorporated into DNA. This is going to be our first introduction to seeing an electrophile that's not a carbon atom, because we're going to be doing some chemistry with phosphate groups. All right, and you've seen that in the pre-video, but you're going to work with it in a slightly different circumstance today. And we're actually going to look at one of, I think it was the first commercially available AIDS, HIV drug, which is AZT. All right, so AZT today. We'll learn how AZT works, and we'll learn why drugs like AZT and a lot of other like cancer drugs really suck. We're going to learn that out today. We're going to learn about why that is today. Okay, again, we have the exam 2 a week from today. We have a review session a week from yesterday. Same bat time, same bat station. Again, I'm open for meetings. So just email me. The time before class is fair game, as well as office hours. If you just give me some notice, we can meet in here, or we can go up to my office to avoid potential distractions. But usually I aim to get to work around 8 on class days. And usually I'm here hanging out 8:15. You're more than welcome to come chat me up, ask me questions either formally or informally. My goal is to get you the quizzes back by Monday. And then I would say have the corrections. We'd have the corrections due with the exam on Thursday. I will post a key and potentially a video where I walk you through what's going on. If you want more practice with problems that are like on the enzyme quiz, I suggest you look at practice exam 2 if you haven't looked at practice exam 2 yet. By the way, speaking of revisions and exams, there was a little bit of confusion about the true-false questions. And I apologize for any inconsistency between the key and my grading, or between the practice exam key and my grading on this exam. I recently changed my policy this semester on grading true-false questions, because I noticed towards the end of the semester, students were just putting not, or not really correcting the sentence in the way and it was as a function as it got more and more as the semester progressed. So I decided to become more strict on that. And I didn't realize that I didn't look closely enough at my old keys to see that they were a reflection of the previous policy rather than my current policy. So this time, when I make the key for exam 2, when I write the exam, I will write them to where I don't contradict myself on the key, and I'll try to make it with questions where you have to answer it with filling something else in. But I apologize. I got several comments about that, and I will be clearer about that on exam 2. But the keys, again, it's a legacy of a previous policy that I've since revised between semesters, and my key just unfortunately does not reflect that. But I'll work on that. But I just wanted to clear the air, set that out now so we can start new for exam 2. Okay. All right. Okay. Quiz is back. And then reflections. I've got your reflections. I've started reading them, but I really want to spend good time with them because there is a subjective element to grading these reflections, and I don't like subjective grading, but sometimes you have to. And so I will probably get these back to you after spring break. Just so you know, for me to get to spend the time that I want with them, I have to prioritize what I'm going to get ready for next week, and it's getting your quizzes back to you and doing a good job writing your exam. But just so you know, you have a heads up front. It's not that I'm ignoring them, it's just they get bumped down in priority. And then lastly, the week after spring break, we're going to be talking about signaling. Those of you who are like interested in neuroscience, HES, disease, neurobiology, whatever, signaling is really cool because that's where we tie in to like hormones and signaling and those kinds of things. It's also where we tie in scientifically to the Dopesick book. So we'll be having our signaling lecture on the Tuesday after spring break, and then we'll be having our paper discussion on the Thursday after spring break. I will post that paper after the exam with a series of pre-questions I will want you to answer beforehand and turn in during class. I may or may not post the discussion questions ahead of time. I have not yet decided about that. But just so you know, that is coming up. And then the nucleic acids quiz will be the 28th. All right. So before, again, we get started, I wanted to take a minute to say thank you for participating and talking openly and honestly and constructively with Dr. McCauley last week. And I wanted to take a minute to address some of the concerns and comments and feedback that you guys had about this class, how I feel about it, and what I'm going to do to address your concerns to make this a better learning environment for all of you for the rest of the semester. Okay, so first things first. Well, you guys had some concerns about the structure of the class, and one of the biggest things was that you feel like there's a lack of scaffolding in the group work activities that we do in class. This is something that I kind of sensed some days more than others. And so one thing that you'll notice today is when we do problem-solving activities, and admittedly, this and next Tuesday are going to be really the last big days where we're going to be primarily group working anyway. But I've tried this time to build foundational activities that have perhaps more basic skills, and then you build. And I'm also trying to orient you at the beginning of the class so you know the skills that you're going to be using. We're going to start small. How do you memorize the nucleic acids? What do they look like? How do they hydrogen bond? We're going to tie it back into ATCAs because I know that was confusing for a lot of you guys. And then we're going to look at what happens when you incorporate the wrong nucleic acid into DNA, and then how can we target nucleotide incorporation for drug discovery. So that's the general path that we're going to be following today. Some of you guys, so again, I'm working on it. So let's see how today goes. I'm hoping that today you'll feel better supported in terms of the activities that I've written. This is also one of the better activities that I've written. At least in my opinion. All right, some of you have observed that the keys for the assignments, especially the pre-assignments, perhaps aren't as thorough, and you're confused and not getting the information that you need. And so one of the ways that I've immediately addressed the problem for exam 2 is I've posted videos where I walk through the key. So hopefully walking through the key, explaining my logic, will be helpful to you guys. And some of it we'd already talked about in class, so some of it may be redundant, but it's there again for you in another form, probably spun it in a different way. And in the future, I'll try to make more detailed keys when we have pre-assignments, and if applicable, if I feel that it's necessary, I'll make the videos. And watch the videos, and if they're helpful, just tell me if you want to see more. There's also some confusion about group roles, and particularly about the role of the reporter. So one of the reasons why we have the reporter is because you guys are doing group work. I'm going around, I'm looking at your work, I'm trying to get a sense for what's going on, but I'm kind of bouncing around and doing a bunch of different things. So sometimes I don't catch everything that's on your worksheet. And so for me, it's really a way of helping correct any misconceptions that might arise upon the way. And a lot of times for this class, we've checked back with the class and I've shown you the answer or we've shared the answer. And so you've gone back and you've corrected your keys and you're good to go. But sometimes things slip through the cracks. And so having the reporter turn in a picture of the notes between two activities is really helpful for helping me figure out where you guys are and then how I can help you and correct anything that may have gone off path. So that's the point of the reporter. And there's also been some confusion about why we rotate group roles. So the format that I take for this class is an adapted of process-oriented guided inquiry learning, or POGLE. Do any of you guys have experience in like a POGLE setup of a class? Okay, a few of you, but most of you have not. So one of the reasons why we have roles within the group and why you guys evaluate each other at the end of each group is because many people feel that in order for you to be invested in group work, you've got to have a job to really invest instead of just sitting and doing it on your own. So that's why we have roles. One of the reasons why we rotate roles is because, and why you sometimes end up with a job that isn't to your strength, is to avoid, so first things first, I'm trying to think of how I can explain this in a good way, and I don't know why all of a sudden I'm getting broken up. But let's see. I'm trying to think of also a diplomatic way to say this. So it's good to give everyone a chance to try out different things. First things first, it mirrors the workplace. So when you're in a job, you're not always going to be asked to do things you're good at. So you have a different chance trying out different things. It also prevents people from dominating the dynamic of a group in a negative way. So if you automatically have the most talkative person being the reporter, other voices in the group might not be heard. But also, instead of arbitrarily assigning roles to and then having you have the same role throughout, it gives people a chance. You don't get stuck with one role that you don't like the whole time. So it ensures that everybody has a chance to try out a different set of skills. One person doesn't take one role and someone doesn't get shafted with another role that they don't like the whole time. And it also gives people a chance to exercise their different strengths. It also forces you to communicate a little bit. So if you're not very talkative and you don't really like being the reporter, your friends who do like to talk or who are better at public speaking can kind of help you with strategies for reporting or something like that. So that's why we rotate. Part of it is because that's the way POGLE is set up, but also I like that you have chances to do different things, and some of the roles carry a larger burden than the others. And so it's good to rotate it and distribute it out. And then lastly, a few of you guys really want to lecture instead of group work. That's okay. There's always people who learn better through lecture. Some people learn better through lecture. Some people learn better through group work. And so that's one of the reasons. And so we have a diverse group of students in this class. You guys come from different areas, different backgrounds, different ages, whatever. So there's diversity in the classroom, but there's also a real diversity in how people learn. And so that's one of the reasons why we do this class. So oftentimes we do problems in class, but there's a reason why I don't just give you book chapters and say read. When I was in graduate school, I had colleagues who did all of the research for their thesis work by reading. And if I had a question, they'd just go, oh, read the book, like I read the book. At that time in my development, I had a hard time getting what I needed from reading a book. And I really benefited from talking to someone about it. And so some of you can read the book and you're fine. That's why I assign book chapters. Some of you need to hear it in a lecture format. That's why there's always a video if we're doing group work. So no matter what, when you're studying, you can go back to the lecture on the video form and get the most essential points from the PowerPoint slides and from what I'm saying. If you're an auditory learner, you get it that way. And then if you're someone who, if I was an undergrad, I perhaps would have defaulted to preferring lecture, admittedly, at that point in my life when I was there. But I also didn't have any classes that were mostly group work, so I wouldn't have even known that was a thing. all of my classes were more traditional, or they were more traditional where we had one day devoted to problem solving outside of class. But some of you benefit from working through problems. And I like the idea of being able to give you things to work on in class, to work together, to parse it out. And so that way, all these different types of learning are being facilitated in some way. Now, the last third of the semester, we are going to shift more towards lecture with problem-solving activities. So those of you who do prefer lecture and still prefer lecture, you're going to be happy the last third of the term. But I'm going to do my best to make sure that we're still doing some kind of problem solving along the way, so you're practicing those skills. And then lastly, some other things. So the elephant in the room. If there was anything about your guys' review or you guys' experience of learning in this class and things that I needed to stop, it's that I talk fast. I know that I talk fast, and I'm really sorry. And it's usually either I get nervous about not having enough time, or I get really excited because I love biochemistry and I start talking really fast. But the past couple times I've gotten reviews from students, I haven't gotten a "you talk too fast," so I guess I've just forgotten that I talk too fast. I don't know how, but either way, I've been reminded that I need to slow down, and so I will try to slow down. And then another thing is that you guys really don't like hand raising. I didn't know that. I guess I should have read your faces. And I did kind of feel like they really don't like raising their hands. So one of the things that Dr. Macaulay, and again, this is a thing that is in the POGIL method. But just because I learned it at the conference from the POGIL people doesn't mean we have to do it, right? We can adapt. So one of the things Dr. Macaulay suggested was clapping. So I clap twice and you clap back. I also thought about, well, maybe I should just yell out Marco and you can yell out Polo back. I think it's kind of goofy. So do you guys think that yelling Marco Polo is acceptable? Do you think that's dumb? Would you rather clap? Does anybody have any feeling about how this is? Any preference? Oh, I'm putting you guys on the spot and you don't want to share. Yeah, Hubert. That's true. Oh, I got to put you on your feet. Okay. All right. Well, so we'll say clapping and I'll yell Marco. I don't know. We'll feel. I'm probably going to lean more towards Marco because I like talking. But either way. And then remember, this classroom isn't a democracy. I'm in charge. However, I'm not here because, yes, I love biochemistry, but I'm not here for my own benefit. I'm here for you guys. I'm here to help you learn. I'm here because I'm passionate about helping students learn. If I didn't care about students learning, I would have gotten much more lucrative industry job right out of graduate school, frankly, okay? So remember, I'm here to listen to your constructive feedback. Even though I'm in charge, you guys do have some agency in your learning. You're not going to debate with me about what goes on the exam, but if something that I'm doing isn't helping your learning, it's okay to give me feedback as long as it's constructive and you're not calling me expletives, which will make me upset. So this kind of segues into my last point, and that this is the role of the reflector. So this is why I pass out reflector sheets with the same 2 stupid questions on them, is because I want to know how your group is doing, but I also want to know what went well, but what really was unclear about the class. And so this is your time and place to give me feedback. So yes, if things were confusing, but if you really talked too fast on that video for the pre-lecture, I mean, you might want to word it a little more diplomatically, but that's your place to put that too. And that's your place. You can also talk about, you can give feedback on how the class went, the pace, all those things. This is the reflector sheet. And just because you're not the reflector doesn't mean you can't contribute to this. So if you're not the reflector, just talk to the reflector and be like, hey, I think, could you put XYZ on the reflector sheet? You can do that. Okay, so I've taken about 20 minutes to talk about this. Before we move on to nucleic acids and nucleotides, does anybody have any comments that they would like anybody have anything they'd like to say? Yeah. Okay. All right. Well, if there's anything you'd like to say you don't want to share with the class, put it on the reflector sheet, talk to me. And then lastly, again, I'm here. I'm happy to talk before class, after class, during office hours, meetings during the week, problem session. Okay. All right. So we have the problem sheets, and I should probably upload my sheet to my PDF annotator. I hope I have a PDF copy. If I don't, I'll make one. Oh, okay. So we're going to make this into a key. It's not a key yet. Okay. All right. So one of the things you guys did on your pre-assignment, I guess we can open up the pre-assignment too while we're thinking about it. Alrighty. And I'll come around and I will look at your pre-assignment like always. Again, it will be a key. It's just not quite there yet. Okay. One of the things you guys did is you drew the nucleic acid because you have to know what the structure of nucleic acids for class. But a really important thing about nucleic acids is how they come together to form hydrogen bonds, because that's the basis of our DNA, which is the basis of our mRNA and our proteins and the things that literally make our body pull together and function. So the first exercise, so there's a lot of memorization that has to happen there. So the first exercise, I want you to talk to your group mates, and I want you to share any ideas or strategies that you may have come up with that will help you keep the nucleotides and the nucleic acid, the nucleotide straight. So go ahead and talk about that for a second, or a minute or two, and then we'll share with the class if you have any strategies. You can draw this, that's okay. I mean, if you look at like the acronyms, they're all essentially very You don't have to, if you feel like it would help you, and you'll want to make sure that you draw, okay, you got a phosphate there, so yeah, make sure you draw it as like PG
I've heard that before. Let's cut the point.
But yeah. There's no other word.
Usually gap, you don't know which key you're referring to. These are four keys.
Okay, all right. So do any of our groups like to share strategies for how they remember nucleotides and nucleic acids? Okay, Rupert. Ooh, that's a good one. Okay. Pure as gold. So adenine and guanine are purines. And so by default, cytosine, thymine, and uracil is your RNA. So again, thymine is the DNA version and uracil are the RNA version of the same nucleotide. It's just that thymine has, okay, thymine has the extra methyl and uracil has that methyl removed. Okay. Any other mnemonic devices? Anybody else? I saw some people writing things down. So one that someone came up with last semester that I thought was, I would say it's a little bit of a stretch, but I thought was clever, is that somebody, to remember the pyrimidines for C and T, they thought of pyramids, and the Egyptians worship, I think they worship, they esteemed cats. And so that's how they remembered C and T, pyramid, pyramidine, Egypt, cat, CT. So again, yeah, maybe that works for some people, maybe it doesn't. But as long as you've got a mnemonic device to remember one set, you should be able to remember the other. The way that I remember, oh, oops, this is definitely on the pre-assignment, not on the, whoops. Okay, I'll fill that in properly while you guys are working on the next problem. But the way that I remember purines and pyrimidines is that, well, this is prior to doing 23andMe. My initials are AG, Annalise Gorensek, and I'm 100% Slovenian, at least 2 generations back, I thought. So I'm pure one thing, and so purines, that's how I remembered it. But I'm not 100% Slovenian, as 23andMe told me. That's okay. I didn't think I was, but that's just how I kept it straight. So, okay. All right. So, we talked about mnemonic devices for remembering your nucleic acids. I will post a key with all the nucleic acids where I have all the numbering, so you can check yours against it. You can also check it against the video, I suppose. Actually, let's take a minute and let's look at the nucleotides and nucleic acids here. Again, our pyrimidines are our 1-ring ones, so our C and our T. Our purines are the 2-ring ones. And so the numbering for the base, for the pyrimidine, the number 1 starts at the end where it connects to the ribose ring. Because again, so it connects to the ribose ring. Whereas the purine starts, oh goodness. If we were to flip this out, let's see. If we were to flip this out, this is the edge where the hydrogen bonding is happening, and we start at the top right, or I guess in this case it's the top left. We work our way around one signal ring, and then we hop over and work our way down, and we actually end on the nitrogen that is connected to the ribose ring. So on the pyrimidine, you start where the ribose ring is connected, and on the purine, you end where it's connected. And again, just as a reminder, those bases are connected to the ribose ring sugar. So this is a ribose. So would this be a ribonucleic acid or a deoxynucleic acid? This would be a ribonucleic acid because RNA or ribonucleic acids have this 2' hydroxyl and DNA does not. And so for sugar numbering convention, we start where the base connects and then work our way up the sugar. And again, this is a ribose. So ribose is a hexoketose, a ketohexose. So it has 6 carbons, but it's a 5-membered ring. So its carbonyl carbon must be a keto instead of an aldose because we have that one hanging off there. Or 1, 2, 3, 4. No, I spoke wrong. Ribose. Rewind. Ribose is an aldopentose because we have 1, 2, 3, 4, 5 carbons, and then it makes a 5-membered ring. Go back to the video. I'm going to delete that out. Okay. All right. So we've really quick touched on sugar numbering. I'll put that nicely in the key for you to go back and look over. So let's move on to our next question on the in-class assignments, which is talking about the ratio of DNA bases. So one of the big discoveries, so back in the day, like in the 1950s, and in Dr. Alexander's class, you actually read Watson's account of discovering the structure of DNA. They didn't really know what DNA was. They knew they had bases. They didn't know how they were arranged. But one of the seminal findings that really helped Watson and Crick solve the structure was this guy named Erwin Chargaff, who discovered that A and T and C and G occur in roughly a 50-50 ratio. Based on your knowledge of nucleotide structure, explain why A and T and C and G must occur in a 50-50 ratio, or a 50-50 or a 1-to-1. So talk amongst your groups and come up with an explanation why Chargaff's discovery must be true based on hindsight, what we know about nucleotides and nucleotide structure.
I just assumed.
Okay, so does any group have an explanation for whether equal amounts of A and T and equal amounts of C and G, in hindsight, with our knowledge or current knowledge of DNA structure, thanks Watson and Crick, but actually Rosalind Franklin, why does that have to happen? Would any group like to offer an explanation? Okay, I think Alex, your hand went up first. Would you agree with that explanation, Caroline? Okay, yeah, so that's exactly it. So one of the things that helped them solve the structure was they didn't know that A and T and C and G complementary base paired to each other. But it was that information, knowing that A and T and C and G had to go together, Watson and Crick were able to figure out and then put those two together and see that they matched up. So we have thiamine and adenine, and then cytosine and guanine here. And so in the exercise, you circled the groups that were capable of hydrogen bonding. And so what you should have seen is that thymine and adenine are both capable of hydrogen bonding with two groups. So in theory, thymine could also wobble, could also in theory hydrogen bond with that guy there, but it doesn't line up with the complementary one on adenine. Whereas if we look at guanine and cytosine, they each have three groups that can hydrogen bond. Now, sure. Angstrom. So angstrom is 1 times 10 to the negative 10 meters. And yes, so it's really important that the distance between these bases works out to be about the same, because if they weren't about the same distance, what happened with your DNA effect, where you're gonna get bulges and tapers and bulges and tapers. And you don't want bulges and tapers, you want the DNA to be uniform, and they were seeing in the structure that the structure was uniform. And so for DNA to be a uniform width, so to be a ruler instead of a strand of pearls, you had to have the bases pairing up with each other to be lined up. And One of the things I regret about not having you guys read the double helix is it goes through before Watson and Crick got here. If you look back, you're like, oh, it must be so obvious that this was the structure. But like Linus Pauling had either a 3 or a 4 strand structure with the bases hanging out and not interacting with each other. So there's all kind of weird stuff that got proposed for DNA before Watson and Crick worked this out based on Rosalind Franklin's data. So, okay. All right. So yeah, so it does matter. And it's what keeps DNA the same uniform width all the way down. Okay. All right, we'll discard those annotations. Okay, so let's then move on to the next question, which is okay. So we've established that we have to have equal amounts of A and T and equal amounts of D and C because they hydrogen bond with each other. However, the ratio of A and T and C and G comes back to become really important for the activity of an enzyme that we've talked about in enzyme regulation, which is ATCase. So I want you guys to talk about how, in your groups, about how the ratio of these nucleic acids affects the activity of ATCase. You can pull out your enzyme regulation notes, your book, whatever, and talk about that amongst your group. And let's connect our knowledge now, our refreshed knowledge of nucleic acids back to what we talked about a previous day in class. Yeah, I'm gonna have to leave that out.
You thought of it that way. You can't have more.
Yeah.
I really wanted a Liberty of London one. I want Liberty of London everything. So really quick, while you guys are discussing, I just wanted to go back and remind everyone of what ATCase is. That'll help. So ATCase is the enzyme that is the first committed step in the biosynthesis of pyrimidines. So let's chant as a class. Which nucleotides are pyrimidines? C and T. So once aspartate takes carbamyl phosphate and aspartate and then turns them into N-carbamyl aspartate. Once carbamyl phosphate and aspartate make N-carbamyl aspartate, there's no going back. That N-carbamyl aspartate is being turned into a CTP or a TTP. Okay? So, and then we talked about some of the ways that the nucleobases regulate it, but I'm going to let you all come to that conclusion on your own. But I wanted to refresh just in case you forgot what ATCase is. So, carry on. Sorry, I was looking over your shoulder. Wait.
like presence of A and G. Like companies.
I'm so sorry. Are you guys doing it? Okay. Margot! Okay. All right, let's come back to the class and see where we are. So in one of my groups, like to start us off and unpacking why the ratio matters for the regulation of ATCA's activity. Yes, okay, Rupert. Okay.
So like so if we increase the concentration of like during like then that was saying like to synthesize more proteins of CMC.
Ah, okay. So if our pyrimidines and our purines are higher, so if the pyrimidines and the purines are base pairing with each other, so they're forming complementary bases. So again, as we see up here, A and T and C and G, ATCase is making pyrimidines, synthesizing pyrimidines. So if there's a high concentration of CTP hanging around in the cell, and by extension, TTP, if we're assuming we're talking about DNA, not RNA, what's going to happen, what this means is that if the cell is going through synthesizing DNA, if there's excess CTP hanging out, that means that there's more CTP than GTP, and by extension, more TTP than more ATP, because they're not getting paired into the DNA. So if we have excess CTP, that means we have too much CTP and TTP, or too much pyridines relative to the purines. And so that's why CTP goes back and inhibits ATCase, because it says, whoa, we have so many pyrimidines that we've run out of purines to match with them. So you need to hold your horses, slow down, and let the cell catch up to the level of pyrimidines that are there. You need to let the biosynthetic pathways for the purines catch up with that for the pyrimidines. So that being said, would anybody like to explain, did any of the groups talk about the effect that ATP had on ATCase? So we understand why CTP deactivates ATCase, but why would ATP activate ATCase? Okay, would you like to elaborate a little bit more? Exactly. So ATCase is kind of like one of the guardians of maintaining the 50-50 ratio, okay? So if you have too much ATP hanging out in the cell, which ATP is a little bit funky because ATP and GTP can get diverted for energy, but we're not going to talk about that right now. But if you have too much ATP hanging out in the cell, that means there's not enough CTP and TTP to keep up with the purine production. So what ATP then does is if there's a lot of it hanging around, it goes to CTP and says, hey, ATCase, upregulate, let's make more. We need to make more CTP and more TTP to keep up with the excess supply of the purines. Yes? Yeah, so if there's too much purine, ATCase needs to make more pyrimidines to keep up with the excess of the purines. So that's why ATP is going to go and turn on ATCase to make more pyrimidines. Yeah, ATCase is making more pyrimidines. No, because if you have too much ATP is a purine. So if you have too many purines, you need more pyrimidines to base pair with the purine. Okay. Yeah. And so that's why ATP goes to ATCase and says, hey, make more CTP to match with the GTP and make more TTP to match with the ATP. And that's how that works. Okay. So, and then I was going to say, I talked about this in the video and the supplemental video for ATCase, which you should check out. But one of the things I think that was a little unclear from the regulation lecture is how altering the Km of the enzyme can have an effect on the velocity or the production of a reaction. And so our Vmax, so let's look at this plot of ATCase activity. The Vmax is about up here. If we go to 1/2 Vmax or the Km and we extrapolate to where the Km is, whoops, no, actually, let's do the other way. So if we pick one concentration, so the Km of ATCase on its own is probably about right there. So if we go to this point on all of the graphs, the corresponding velocity of that the velocity of that enzyme at that substrate concentration is at about 1/2 Vmax for ATCase alone. But when we add CTP, which raises the Km to slow down the enzyme, at that same concentration, you're at a lower velocity than you would be at that same concentration if you just had the enzyme alone. Similarly, ATP is binding to ATCase, and both of these are making a change in the conformation of the enzyme that changes its Km. So if we have ATP binding, it's allosterically changing the shape of ATCase in a positive way, so the Km is becoming smaller. And when that happens, if you go to that the velocity of that same substrate concentration, it's increasing. And so that's how by shifting the Km, you can increase the productivity because you're lowering or raising the velocity of the reaction and therefore the production of the product at the same concentration of substrate. So like in this case, we would be thinking of this as the cellular concentration of aspartate. I don't know what the cellular concentration of aspartate is, but I picked that place on the plot to illustrate a point. Okay, are there any questions before we move on to the next question? Okay, all right. So we know why we talked about base pairing. We understand base pairing leads to the 50-50 ratio. We've tied the 50-50 ratio back into our previous work with enzyme regulation and hopefully gotten a little bit of a nice refresher for the exam. And so now we're going to talk about what happens when things go wrong with when things go wrong with hydrogen bonding, and what does the effect have on the structure of the DNA. So you guys have probably heard at some point or another that going to a tanning bed is a bad idea, No offense if any of you are tanning bed people or standing out in the sun without sunscreen, or for me, it's just going out in the sun because I burn even with SPF 50 is a bad idea. Because when you do that, it can alter the structure of your DNA. And so one of the things that can happen is you can deaminate the structure. You can deaminate cytosine, and when you deaminate cytosine, it turns into uracil. So if we look to have the structures of the nucleobases on here, so cytosine right here, we deaminate it. So the amine is swapped out and replaced. The amine is replaced with an oxygen. And so that's one of the reasons why going out in the sun is bad, because UV light can turn your cytosines into uracils. So what you're going to do now is you're going to work in your groups to see why this is a bad thing. What happens? So what? We turn one into another. What does that mean for our body? Why do we care so much? So we're pretty close to this one. So essentially, those positives can be filled by different enzymes. This is present. So they're all. No, so it makes so actually decarbonyl. It eventually shuttles on to faster. It'll get these things that will get turned into CTP or CTP. Yeah. So in this case, ATP on start ones. But GTP wouldn't have to. The groups in the binding and the allosteric binding of ATCs. Yes. So Rishon just had a really good question. He was wondering, if ATCAs made CTP and TTP equal ratios, and also if ATCAs could bind GTP. So first things first, ATCAs is making a precursor for CTP and TTP. So there's going to be a point where that precursor is going to be diverted. Some of it will go into CTP and some of it will go into TTP. And the ratio of CTP to TTP, you can look it up on the internet. It's close to 50-50, but it probably isn't quite 50-50. Some organisms are biased more towards C and G, and others are biased more towards A and T. So it just depends. But I want to hit home that it is a precursor. It's making the first product that will turn into CTP or TTP. It's not associated with the downstream. And the second one is that GTP and TTP don't regulate ATCAs. The allosteric binding pockets would be such they wouldn't recognize those nucleotides. However, we can think of those as being a proxy for the whole pool of purines and pyrimidines respectively, and that's exactly what's happening. Nature was lazy enough that it evolved ATCAs to recognize one purine and one pyrimidine. Carry on.
North Wind to come.
it's just the one condition.
Switch. H2 used to be a hyphen. So and then like as hyphen used to be the oxygen is a hyphen. Except it's
Oh, rats. I thought I turned off sharing.
Wait,
students. So not everyone has fully drawn out the nucleotides and the hydrogen bonding. That's okay. I highly recommend. So you're going to have to memorize the structures for the nucleic acids quiz. You're also going to need to know how to draw on the hydrogen bonds. So I highly recommend you practice drawing 2 complementary nucleotides and drawing in the hydrogen bonds on your own time. And this works best if you flip one or the other. So here, in my lovely chicken scratch drawing, because I was trying to write quickly, we have the hydrogen bonds between C and G. So you can see C and G again have 3 hydrogen bonds, and U and A have 2. Okay, so when you have UV exposure to your DNA, one of the things that can happen is the C can be turned into the U. Now, this happens. This happens in our body. Sometimes our polymerase puts in the wrong base, and so our body has developed systems to find these incorrect bases, nick them, and pull them out and repair the DNA. When trouble can really happen is when those repair systems don't catch the errors. And so the question that I asked you is, why is that a problem? And if you need a minute to think about this, I want you to observe, if you swap out C for U, will you be able to make the same complementary base pair bonding? Take a minute as a group. If you were struggling a little bit, now I want you to think in terms of C, make the same hydrogen bonding pairs as U, and how would that have an effect on our genome? All right? Let's break. I apologize. I don't. All right, Marco. Okay, guys. All right, tell me, can uracil hydrogen bond with guanine? Can it? Yes or no? Shout it out. No, it can't. Well, okay, so if you look at the faces of those two, and if we're to look at a different color, you have an oxygen and an oxygen. You have a proton and a proton. You have now the oxygen and the hydrogen coming off of that amine could hydrogen bond, but that's one hydrogen bond when you usually have 2 or 3. So that's not going to happen. So if you have a cytosine swapped out for a uracil, what's going to happen to the genome if that uracil doesn't get removed? Anybody? Some of you have answers written down. Let's see. Are you raising your hand, Grace? Or you're fluffing your hair? Okay. Right. So we're not touching a whole lot on replication in this class, but when your cells divide and you're going to synthesize a complementary strand to the strand that now has the uracil in it, because uracil doesn't hydrogen bond with guanine, instead of putting in guanine in the complementary strand, it's going to put in an adenine. And so if you make a deletion, if it's in like a non-expressing part, or depending on what the corresponding codon for the amino acid looks like, you could change the amino acid that segment of the DNA codes for. You could end up with a different amino acid and a protein, and then you could end up with a protein that doesn't work. That's how you end up with something like sickle cell, where you go from a polar amino acid to a valine, and then you get hemoglobin clumping to each other instead of working. So this is one of the reasons why UV damage is so concerning, because it can literally lead to an alteration in the sequence of your genome. And as we know, DNA to RNA to protein bad proteins, bad problems. So, okay. Are there any questions before we move on to our last question? All right, so we'll go ahead and we'll move on. So in the video, we talked a little bit about nucleotide incorporation, especially because I wanted you to get practice doing chemistry with phosphates, because we're going to see a lot of phosphates in the last term of the last third of the semester. But I also want you to get practice at drawing a nucleotide in the context of the chain in DNA or RNA. So what you're going to do is we're going to talk about an HIV virus viral inhibitor called AZT. So we talked about one inhibitor that inhibits HIV protease, which chops apart the GAG protein and the Pol protein. Today, instead of talking about inhibiting a key protease, we're going to talk about something that inhibits HIV polymerase. So after GAG-Pol's been cut, you get the viral polymerase. Now we're going to talk about a drug that inhibits the viral polymerase. So what I want you to do is we're going to take this incomplete diagram of the viral DNA, or I guess it would be a DNA strand. Okay, HIV. So HIV reverse transcriptase, it takes RNA and makes a DNA copy of it. So what you're going to do is actually, it isn't the polymerase. We're inhibiting HIV reverse transcriptase. Apologies. You're going to be looking at a growing strand of DNA that HIV reverse transcriptase is in theory making a copy of from its own viral genome RNA. And you're going to see what happens to that growing strand if you try to throw AZT in there. All right? So go ahead and let's work through quickly parts A, B, and C. I'll filter through and have her around. If we don't finish it, we'll bundle to the pre-limit for next Tuesday. Right. That's right.
Oh, yeah.
Yes.
So you can manipulate it, and I think there's a picture of it on where you kind of, it's like upside down, you can flip the bases of this matches. Because what's representing two bases, what two DNA is in your space? Okay. What were you asking? Let me pull up a picture.
Oh no, I didn't draw that in. Ah. Here we go. So you can kind of flip the sugars. Just put the sugars upside down. And it's going to come up with practicing drawing it out. But honestly, what I'll do is I'll write in the bases and then draw it. Honestly, that's how I feel it.
Like label the linkage between two types. So is that just drawing the
So you're going to name the bond next. So for part B, the bond that links two nucleotides together, or rather the two riboses together, has a special name. And so I want you to identify what the name for that bond is. If you don't know, that's okay. We'll come together on it, but I want to make sure we know what the name of that linkage is. All right, resume. Well, and so that should help you see the riboses. Flip them upside down. Yeah. Yes. Marco. Okay. All right, let's come back to the front. So we're not going to finish this problem today. That's okay. So I'm going to put this as part of the pre-assignment for next Tuesday. But what we can go over before we adjourn class today, which end of the newly synthesized DNA is the 5' and which is the 3'? Which am I going to put up here at the top? The 5' or the 3'? The 5', right? And then what's the bottom? 3. So if you got a little confused, remember it's always the 3' hydroxyl and the 5', and then the phosphate is 5'. So I always remember it by thinking of the 3' hydroxyl hanging off. And so DNA is always going to have the 3' hydroxyl, as is RNA. The difference is the presence or absence of a hydroxyl at the 2' position for 1', 2', 3', 4', 5'. That's the 5'. That's where the 5' phosphate comes from, and the 3' hydroxyl comes from the numbering on the sugar ring. And again, we use primes to distinguish the numbering from the base numbering. All right? Okay, so that's 5' to 3'. So some of you probably haven't drawn in the RNA yet. So how would the direction go for the complementary parental strand of RNA? Down here, would we have the 5' or the 3'? So we'd have the 5' down here, and we'd have the 3' up here. So we're going to have our phosphate, down here, and we're going to have our ribose rings oriented like so. I'm sorry, I'm butchering drawing a pentagon. Going down there, we would have the linkage coming off of the complementary base coming off of there, and then the OH to the next one, and so on and so forth. So that's how you would start to draw in that complementary, is you're literally going to flip the ribose upside down, but you can imagine the DNA base freely rotating around its glycosidic bond, so you could draw it both. And pointing out the bond names, what is the name of the linkage that connects the two ribose rings in this case? Addie. Yes. So this is called a phosphodiester linkage, and that's because you have a phosphate or a phospho, and then you have two esters connecting to the two sugars. So the phosphodiester bond, because you have an oxygen, two oxygens connecting the phosphate to two other atoms. That's where the ester comes in. Okay, so I think that about covers it. So for the pre-assignment, I'll have you draw in that parental strand and propose the mechanism of AZT, and then we'll adjourn talking about nucleic acid structure on Tuesday.